Okay, wonderful. So it is 1 o'clock. I just wanted to say, hello and welcome to the Invasive Mussel Collaborative webinar series. We have some excellent speakers today who've agreed to share their work with us. So presenters David Kramer with the Michigan State University will be sharing his work regarding tools to identify water bodies critical to the western spread of an invasive species. And then Michael Springborn and Joseph Raymond with the University of California, Davis will be presenting their work on the role of key environmental drivers and management scale in assessing spatial variation in Dreissena spread risk in the Missouri River Basin. So some great presentations from the western portion of the United States. My name is Nicole Angel, and I'm joined by my colleagues Sam Tank and Autumn McGowan with the Great Lakes Commission. And on behalf of the IMC, we just want to thank you for participating in today's webinar. So before I turn the presentation or the webinar over to our presenters, I just wanted to give some background on the collaborative. So the Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for the management and control of Dreissena mussels. And this is in order to produce ecological and economic benefits. So the Invasive Mussel Collaborative published a management strategy in 2018 to reduce invasive mussels and their negative impacts. So to make progress on the goals, the IMC's goals, the collaborative established 4 work groups, which you can see on the screen here. So these are the research work group, Dreissena toxicity testing work group, coastal site priorities work group, and most recently established the planning and implementation work group. I'm just going to briefly highlight each of these work groups just to give you a sense of, what kind of work the IMC does. So the research work group is charged with coordinating existing research and developing targeted plans and projects that address Great Lakes management priorities. And this work group released a mapping tool called the Dreissena Project Coordination Mapper, which you can see here on the screen, and you can access via that bit.ly URL. Basically, this mapper allows researchers and managers to sort of share their work and collaborate with others for the advancement and protection of waterways from invasive mussels. And this research work group is actually currently soliciting more research and applied control projects for this mapper, and you can actually just click on the little buttons on the screen here to add any projects that you might have to the mapper. Moving on to the toxicity testing work group. This group was charged with reviewing both the strengths and the weaknesses of mussel toxicity control methods used across various labs to identify and make recommendations to further refine and standardize methods. So these studies reviewed by the work group has been compiled into a table that provides sort of a searchable collection of literature for Dreissena laboratory toxicity and open water control testing methods, and is available on the website. Additionally, a manuscript detailing the recommendations of these different recommendations can be accessed via the bit.ly URL on the screen. Moving on to the coastal site priorities work group. This work group was charged with reviewing information on where Dreissenids are impacting Great Lakes resources and evaluating sites based on this information. So this work group released this interactive dashboard and map that displays nearshore data from the Coastal Site Priorities Index. This tool is called the Great Lakes Experimental Mussel Suppression Site Screening Tool, which is a mouthful. So we just call it GLEMPST, and it features a dashboard and associated indices, which are intended to both inform future applied Dreissena research and management activities and aid site prioritization. And then finally, moving on to the last work group, the Planning and Implementation Work Group, which was charged with developing a research and management roadmap for the site and regional control of mussels. So they are working to identify stakeholders and funding priorities and remaining knowledge gaps to guide future Dreissena control activities. And this work group is really building on the products and outcomes of the other 3 work groups. So you can be on the lookout for more exciting progress from this group over the next coming year. And then moving on generally to the Invasive Mussel Collaborative. So today's webinar is part of a communication network facilitated by the IMC. And the purpose of these webinars is really to facilitate learning and information sharing on topics of common interest. So the webinar will be recorded and archived along with past webinars on the IMC's website. And if you're interested in learning more about the collaborative or staying in touch with our communication networks, we do encourage you to visit our websites and join our listservs. There's lots of great information and ways to connect there. And then finally, getting into some housekeeping, we do strive to minimize tech issues during these webinars, and we apologize for any that might arise. But we have automatically muted attendees just to reduce interruptions, and you'll remain muted with your webcams off throughout the presentations. However, we do encourage you to write your questions during the webinar, during the presentations, using the Q&A function. So questions can be submitted anytime during the presentations. However, if you're experiencing tech difficulties, those go in the chat. So Q&A for questions for presenters, tech, or any other things you want to introduce yourself, say where you're from, put those in the chat, please. And if you're experiencing any tech difficulties, you can also directly message or email Sam, my colleague from the commission. Her email is on the screen there. So without further ado, we'll get to the meat of our webinar today, what we're all here for. And I'm going to introduce our first speaker. David Kramer is a PhD student and fisheries biologist specializing in invasive species research. David earned his master's degree at Texas Tech University, studying the ongoing zebra mussel invasion in the Southwest U.S. He has continued his work on invasive species as a PhD student at Michigan State University, where he develops potential management tools for sea lamprey control. And with that, I will hand it over to David. I will stop sharing my screen. David, you can pull yours up if you'd like. Make sure everything's working, and then I'll turn my camera off and mute as well to give you the floor.
All right, thank you so much for that introduction, Nicole.
Yep, everything looks good from my end. Thanks, David.
Okay, sounds good. Thank you. Yeah, and as Nicole said, my name is David Kramer, and today I'll be talking to you about our recently published paper on the western spread of zebra mussels in Texas and New Mexico. So I'm not going to spend too much time on this, but I did want to give a quick background on zebra mussels to ensure we're all on the same page. So from an initial invasion of the Great Lakes in the 1980s, zebra mussels have rapidly spread across the eastern and central United States, causing great ecological and economic damage in the process. However, they have been less successful in spreading to more arid western regions. And they are a more recent arrival in Texas, only arriving in 2009. And our map here shows the current status of zebra mussels within our study area. New Mexico is still zebra mussel free, while in Texas, zebra mussels have been detected in 35 lakes, with 33 of those lakes classified as infested with permanent reproducing populations. So what can we do with this? What can we do about this? Early detection and rapid response is one of the most common frameworks for anti-invasive efforts. The emphasis on early and rapid is well-founded. As seen in the graph here, as the time since the invasion increases, the cost of anti-invasive actions also increase. We also quickly lose the ability to eradicate the invasive and instead just must control the problem. So we know that anti-invasive actions work best when used as soon as possible after an invasion. However, it is not realistic to monitor every possible habitat in a region that could host invasives. So to most effectively use EDRR, we need to know the where or what specific areas are important to invasive spread and establishment. Now, what is required for zebra mussels to successfully infest a lake and establish a permanent reproducing population? First of all, they have to physically arrive at the lake in question. They have to be introduced. Mussels can be introduced through a variety of vectors, but they are particularly adept at latching onto boat hulls and propellers. They can then be transported to new lakes by the overland transportation of these boats. But it's not enough to just be introduced. The environment must also be conducive to their survival and reproduction. And this is determined by the water quality parameters found at that lake. And when we have those two factors, infestation of a lake is possible. So in our project, which took place in Texas and eastern New Mexico, we used network analysis to identify lakes important to the zebra mussel spread across the study area and habitat suitability indices to evaluate each water body's suitability to zebra mussel establishment. And let's begin with habitat suitability. Habitat suitability indices, also known as HSIs, were calculated using the water quality parameters listed here. The water quality data came from a variety of state and federal sources, from other academic sources, and my own personal measurements taken in the spring and summer of 2022. This combined data set had water quality measurements for 152 lakes in Texas and eastern New Mexico. And from that combined data set, we assigned a value of 0 to 1 to each lake, indicating its suitability to zebra mussels. And these values were determined by the known environmental tolerances of zebra mussels within each of these water quality parameters. And to be clear, the HSI goes from low to high suitability. So a lake with a score of 0 would be unsuitable for zebra mussels, while a lake with a score of 1 is extremely suitable. To improve our water quality data coverage, we also visited 17 lakes in West Texas and New Mexico to make those in-person water quality measurements I mentioned before. Unfortunately, even after incorporating the water quality from these supplemental sites, multiple lakes still had incomplete data sets for parameters of interest. And to input this missing data, we used regional water quality maps, correlation with other parameters, and replacement values such as air temperature instead of water temperature. Now let's move on to the network analysis. The spread of an organism through their environment is often represented by habitat patches and the movement between them. In network analysis, these habitat patches are nodes, and the edges are the dispersal corridors between them. The maximum lengths of these edges is typically the maximum dispersal distance of the organism in question. And this makes sense. If two pieces of habitat are within a dispersal distance, then an organism can travel between them, and those patches or nodes should be connected in the network. In the case of zebra mussels, whose dispersal is so helped by the overland transportation of those boats, the nodes of the lakes, while the edges connecting them are roadways between them. The maximum edge length of the network is then determined by the distribution of distances boaters would travel, according to a boater survey by Texas Parks and Wildlife. In general, the 95th percentile of dispersal distance is often considered the maximum dispersal distance for a population. Thus, that distance is the focus of our results. But please note that we created networks of the 50th and 75th percentile of those boater travel distances found in that Texas Park and Wildlife survey, when we created these networks for comparison. This resulted in 3 separate networks using maximum edge lengths of 363, 125, and 51 kilometers, corresponding to the 95th, 75th, and 50th percentiles, respectively. So we have built our network in ArcGIS. What do we do next? Well, network analysis allows us to create centrality measurements to describe the connectivity of the network. And let's take a look at these centrality measurements in a very simple network. And again, imagine each one of these nodes represents a lake. Our first measurement was the degree scores, which is the number of edge connections a lake has to another lake. High degree scores means that lake is a hub within the network. So what lakes here have the highest degree score? Well, we have 2 different lakes that are each connected to 3 other lakes, so both of these would be hubs within the network. Betweenness on the scores, on the other hand, are the frequency that a lake will be located on the shortest path between other lakes. In other words, a lake with a high betweenness will serve as a stepping stone from one area of the graph to the other. Now, in our example here, where is the highest betweenness score? In this case, it is the red lake node here because 3 different parts of this network, which I've circled in purple, have to flow through this node to reach the rest of the network. Thus, it is a stepping stone. Finally, we have cut points, and cut points are simply nodes that, when removed, increase the fragmentation of the network. So any lake here that is removed from the network then splits the network into two disconnected parts. Finally, we use both our network analysis and HSIs to identify lakes critical to the spread and establishment of zebra mussels. First, we need to actually identify the cut points, hubs, and stepping stones. Cut points are simply identified in the iGraph package within R, no further work needed. But for hubs and stepping stones, iGraph gives us just the degree and betweenness scores for each lake. It doesn't actually identify what exactly we'd consider a hub. So we considered hubs, those lakes in the top 20% of degree scores, and we considered stepping stones to be in the top 20% of, we identified lakes as hubs if they were in the top 20% of degree scores. And then we identified lakes as stepping stones if they were in the top 20% of betweenness scores. And finally, we must note that any lakes considered unsuitable for zebra mussels by the HSIs could not be considered a critical lake. No matter how well positioned a lake may be within a network, if it is utterly unsuitable for zebra mussel survival, it will not act as a hub or stepping stone. So let's explore our results. First, with the Habitat Suitability Index. Dark brown is no suitability for zebra mussels, blue is low suitability, yellow is moderate suitability, and orange is high suitability. As you can see, outside of East Texas, almost all the lakes of our study area have a moderate to high suitability. The concentration of no and low suitability for zebra mussels in East Texas is due to suboptimal values for pH and calcium in the region. I see my camera is off, but I haven't heard anything. I assume my presentation is still being heard, so I'll keep going.
Yep, looks good.
Okay. Now let's move into the graph theory results. For the sake of time, I'll only be discussing the centrality scores of the 95th or 363 kilometer percentile network. But as we will see later, all three networks will be considered for the critical lakes. And in the appendix of the paper we just published also included every single network we made. So the degree scores are divided up by quintiles. So dark brown is the first quintile or the bottom 20%, and so on. Degree scores were highest in the northeast part of the state, particularly the Dallas region, and declined as we moved further south and west into more arid areas where our lakes became more isolated. And then, like degree scores, betweenness scores were divided up by quintiles, and between scores varied greatly over the study area with no obvious geographic pattern. And as for our cut points, we have a blank map. At 363 kilometer edge length distance, we actually did not find any cut points, which has implications that will be discussed later. So here we have the hubs, cut points, and stepping stones found at each network. And we have the 51 and 125 kilometer edge length networks on top, and the 363 kilometer network on the bottom. And there's a lot to discuss here, but let's focus on a few key points. In all three networks, hubs, or the orange crosses, were concentrated in northeastern Texas. Now, in the highly connected 363 kilometer network, there were no cut points present, and cut points only existed at the lower edge length networks. Finally, stepping stones in the 51 kilometer network had very high overlap with hubs. However, as the edge length increased, stepping stones moved further south and west in the study area. In particular, these stepping stones in West Texas and eastern New Mexico could potentially facilitate the further western expansion of zebra mussels in the study area. Now, what are the implications of this work? Well, outside of East Texas, the study area is broadly suitable. However, while we did find the water bodies in West Texas and New Mexico to be suitable, there were clearly less of them, leading to water bodies more isolated from each other, as shown by the degree scores. So aquatic habitat is rarer in West Texas and New Mexico, but the habitat that does exist is mostly suitable. We can use graph theory and a gravity model that I'll discuss later to determine if the rarity of aquatic habitats will serve as a dispersal barrier to zebra mussels. Let's discuss those graph analysis results. First of all, when using a commonly accepted definition of maximum dispersal distance, that 95th percentile, we had a highly connected network without any cut points. In ecology, a fully continuous network suggests that the organism in question can disperse throughout the network. We not only have a continuous network, but even if we remove a node, the network stays continuous because flow can continue through other connections. These results strongly suggest that zebra mussels have multiple pathways of dispersal through the network. Then our hubs represent lakes that may serve as a source of further zebra mussel spread in the network, while lakes identified as top stepping stones may be those critical lakes that zebra mussels use to expand their range throughout Texas and into New Mexico. Now, coming back to our EDRR framework, managers can guide the allocation of limited time and resources to preventative campaigns like the Clean, Drain, Dry campaign seen here, and monitoring activities testing for the presence of mussels. Importantly, water quality and network analysis measure different things and can serve as complementary data sources informing the decision-making process. Furthermore, our results identify lakes that can still be targeted for management activities long after infestations. Actions at infested lakes are still important as a means to prevent further mussel spread. Finally, we were forced to rely on data imputations for some lakes, as I briefly touched on, and some lakes could not be evaluated for establishment risk at all due to lacking any water quality data. We suggest that further water quality collection should be targeted to lakes scoring highly in our network analysis, as if these lakes are in fact suitable, they may play an outsized role in mussel spread. And that concludes my presentation on our recently published paper utilizing HSIs and graph theory. And as part of my thesis work, I also create a gravity model to simulate the movement of boats in the study area and calculate risks. Now we don't have this published yet, but I wanted to give a quick overview of this tool to wrap up our presentation. So what exactly is a gravity model? It is a tool for estimating the movement of various entities within a network to various destinations. And in their simplest form, gravity models just need the distance between an origin point and a destination and the attractiveness or gravity of the destination to whatever entity is moving. We can also define the number of entities at each origin point prior to them dispersing through the network. So how does that work within our context? We looked at boater registration data and considered the origin point for boats to be the zip code they were registered in. Then we can simulate the number of boats moving from the zip code, the origin zip code, to the lake destinations based on the attractiveness of those lakes to boaters and the distance between the lakes and the zip codes. Then to represent the establishment risk, we incorporate our habitat suitability submodel, which is we have already discussed. Our model produces estimates on the number of infested boats that visit each lake and successfully introduce zebra mussels. If this exceeds a threshold value, then the lake is infested, and the results of one model iteration are fed back into the model to simulate the spread of zebra mussels year over year. Finally, we varied several additional parameters, as we will soon see on the next slide. So here are those parameters that varied between model runs. The maximum boat a distance could travel to reach a lake destination within the network in kilometers, a distance decay coefficient, and the threshold of infested boats needed to infest a lake that we discussed earlier. Each model run selected from the possibilities here, and in total, we ran 2,240 model runs with unique parameter combinations. We examined the influence of many of these varying parameters had on infestation predictions, but for this presentation, we'll be just focusing on the influence of maximum distance in kilometers had on model results. Our next objective was then to evaluate the gravity model and to determine the best performing model runs. And we evaluated the model results against the current infestation status of Texas lakes in order to do this. Now, at this point, you might be feeling like our friend here and wanting a reminder of why we are doing this in the first place. So there are many unknowns with any gravity model, and ours is no exception. So it wouldn't be a good idea to just pick one value for the maximum boat or travel distance, for example, and just report the results of a single model. Rather, what we have done here is include many models with varying parameter values to generate many results. If a lake is frequently infested across different versions of the model, those lakes are likely at a high risk of infestation. So we determined the proportion each lake is infested across all best performing model runs. This also allows us to evaluate the influence of specific parameters, such as max D, on the infestation patterns found in the model. Now let's talk about our model results. After evaluation, we selected the 634 best performing models out of the 2,000 plus models we performed. Here we have the proportion each study area lake was infested across all best performing model runs. And the orange dots here are our most frequently infested lakes. As you can see, these are concentrated in Central Texas. Infestations outside of Central Texas were relatively modest, with very low infestation proportions in West Texas and New Mexico. And here we have an animation showing the results of one of our best performing model runs and the years each lake was infested. Please note that many lakes were never infested and are colored gray. In our best performing model runs, new infestations decreased over time and sometimes stopped prior to the 2040 end date of the model. In fact, only 144 of 634 best performing models had new infestations after year 14 or 2022. And in our example here, steady expansion takes place from year 1 to year 11, but further expansion simply stops and no further spread takes place. Now let's look at the influence varying max D had on model results. At 100 kilometers, map A, the highest rates of infestation were in Central Texas, and infestations outside of this region were very rare, with no infestations in Central Texas, in New Mexico, excuse me. When we increased this max distance to 200 kilometers, many lakes outside of Central Texas faced higher infestation proportions, and a single lake in New Mexico now faced infestations. Now, when we increase the distance to 400 kilometers, the infestation proportions of many lakes outside of Central Texas increased even more. However, infestation proportions at those Central Texas lakes actually decreased, as can be seen by the total absence of our orange-colored lakes, which were lakes infested in at least 75% of model runs. Now, interestingly, if we looked at how often each of the potential max D values were found in our best performing models, we find that better performing models tended to have lower max D values, especially if we looked at the very best performing models or the top 5%. Max distance values of 150 and 200 kilometers were the most common values in the best performing models. And nearly all the top 5% of models used a max distance less than or equal to 200 kilometers. So in summary, the use of lower maximum distances seem to lead to better performing models. Now, what does all this mean? Again, outside East Texas, the study area is broadly suitable. But a cursory glance at the map of our lakes used in our gravity model shows that the lakes become rarer as we move west. So will the rarity of aquatic habitat serve as a dispersal barrier to the mussel? We already showed how graph analysis analyzed this question, but gravity models can provide insight as well. Many central Texas lakes were infested across many model runs, thus are likely at high risk of infestation. However, lakes outside this region faced much lower risk of infestation. The low infestation risk in East Texas is not due to a low introduction risk. Rather, the environmental suitability of many East Texas lakes is low, and the low risk of establishment leads to a low infestation risk. The water bodies in West Texas and New Mexico, on the other hand, were broadly suitable to zebra mussel establishment. However, zebra mussel spread is hurt by the scarcity of these water bodies. As we see here, they just can't find enough water, and they just can't find that habitat. In model iterations where the boats were allowed to travel up to 400 kilometers to reach a destination, these limitations were overcome, and the western portion of our study area did see some lake infestations. But those high max D model iterations performed very poorly when compared against the historical spread of mussels in the study area. This suggests that our boaters really are traveling shorter distances, and thus a invasion front emerging from central Texas encounters less and less potential habitat for zebra mussels as it moves west. So even though the habitat that does exist is suitable, the scarcity of them limits potential sources of zebra mussels to infest boats and engender further spread. Finally, I wanted to end on the potential future directions this line of investigation can take. Applying the tools of graph theory and gravity modeling to invasive species is an increasingly common strategy. And within Texas alone, there's already another invasive mussel, the quagga mussel. In addition, an invasive plant known as giant Sylvania has already caused a lot of damage in Texas. Both of these invasive species are transported by recreational boats, and there's great potential in applying our methods to these species. These are just two examples of how our work can be applied to a variety of invasive species, both within Texas and outside of it. And here are the references for anyone who would like to see that. And I wanted to quickly make note of all the agencies that help make this project happen. Of course, my lab at Texas Tech, and I'd like to help thank all the technicians and my fellow graduate students who helped me with this project, both in the field and in the lab. And my understanding is we're saving questions for the end, but when that happens, I'm happy to answer any questions you have.
Well, thank you, David. Really interesting work. Thank you for presenting. Let me go ahead and I'm going to share my screen quickly. I should be able to see that now. So I did want to remind everyone, like David said, we'll save questions till the end. As a reminder, put your questions for David in the Q&A using the Q&A function. If you go click on Q&A, there'll be a bar that says start a discussion. You can just toggle down and switch that to ask a question if you'd like to ask a question for David. So thank you again, David. We're going to move on to our next presenters. Our next presenters are Michael Springborn and Joseph Raymond. Michael is an environmental and resource economist and a professor in the Department of Environmental Science and Policy at UC Davis. He works on problems of resource management and decision-making under environmental risk and uncertainty using econometrics and dynamic optimization. He mainly works in applied areas of invasive species, endangered species, disease, and fisheries. And then Joseph Raymond is a PhD candidate at UC Davis with interest in environmental and natural resource economics, invasive species policy, and fisheries risk management. So I will end my screen sharing, and Joseph or Mike, if you want to share yours, perfect. Okay. Not in presenting mode just yet. Okay, there it goes. Looks good. I'll turn off my camera and you guys can take it away. Thank you so much.
Very good. So I've got my camera on, but just let me know if I should turn it off for bandwidth's sake.
No, you can leave it on. I'm not sure why David's went off. Some weird glitch maybe, but yeah, feel free to do whatever you're comfortable with. Thank you.
Very good. Okay, well, thank you so much for that introduction, Nicole, and for the invite to be here, to you and Samantha, and also for David for kicking us off with such an interesting project. I'm going to start here, our presentation, and then turn things over to Joe at the midpoint to take us home. I just wanted to acknowledge a few of our other collaborators on this project, mostly from the U.S. Geological Survey, Wes Daniel, Lucas Bear, and Tim Cunahan and Matt Nielsen. As well as then Sophie Duntugan from Northeastern University. This presentation is going to reflect some research that we recently published in the paper that you can see here. We also created a Shiny app tool that we'll talk about later in the presentation that implements the model and gives any interested user control over some parameters of the model. As we get towards the end of the presentation, I'll drop those hyperlinks to the paper and that tool into the chat and share those in case folks are interested in checking that out. So our objective with this project, we were focused on the Missouri River Basin, and we were trying to find a practical metric for prioritizing what should be high mussel risk areas within that river basin for quagga and zebra mussel risk. And there's a lot of challenges, as you probably gathered from David's presentation. There's a lot of information that you want to bring together to try to address this kind of problem. We've got a large and spatially heterogeneous region in the Missouri River Basin. And you can think of lots of different important kind of metrics or concerns that would feed into any sort of sense of a risk or a threat. And that leads to, in our case, about a dozen different data sources that we'd like to bring together. But integrating that data in a conceptually coherent way that's also practical and scalable over the entire Missouri River Basin is obviously a challenge. And even once we have those metrics and we can try to bring them together, the sense of how important one metric is relative to others, what kinds of damage are more important than others. For example, there might be differences in beliefs on the relative importance of those different inputs to the model, and there might just be objective differences in how important those things are as you sort of move across space. So we wanted to try to come up with a coherent framework to address all of these challenges together. Here's a map of the Missouri River Basin, and it includes the Huck 10 level watershed units. Those are the polygons that you can see within the Missouri River Basin. This is, of course, in the north central United States or so, at least from east to west. You can see in the bottom left of the map where this is located within the US. And just to anchor us to this particular basin, what's plotted here is a sense of the water intensity in any given Huck, a combination of both lakes and ponds, as well as the flow going through rivers and streams. So combining those together, what are the relatively water intense watersheds within the Missouri River Basin? And the scale here, the top of the scale is in yellow. These are the most water intense watersheds in the Missouri River Basin. Not surprisingly, you tend to see those mostly focused along the major rivers. Juxtaposed on top of that are the red dots, which indicate at the time that this was created what the existing locations for mussel infestation were within the Missouri River Basin and also just outside of it, as you can see to the east and to the south. So our approach here involves combining several different data streams, which are our subcomponents in light blue kind of around the outside, that all feed into three key components of our composite relative risk measure. So this composite relative risk measure, or CRR, is that ultimate metric that we're going to boil down a single number for each of the watersheds in the Missouri River Basin. It's composite in the sense that it's bringing together these different subcomponents in the mid blue here. And the way that we bring these together reflects this idea that the risk that we're concerned with is a combination of the event likelihood, and the event here is an infestation. So the risk is a combination of that event likelihood, the likelihood of an infestation, times the impact of that event. How damaging or concerning might that event be? So we've got our infestation threat component here in the mid blue, but then also two different subcomponents, or sorry, components, value at risk and spread threat. The value at risk is meant to capture what do we think the relative immediate damage within a given watershed might look like. And that could come from things like what are the different beneficial uses in that watershed that might be at risk. But of course, once a water body is newly invaded, there's another impact that's generated, which is the threat of additional spread, which is that third component that we see down there at the bottom. So those two things, the value at risk and the spread threat, give us a sense of impact. And so we're adding those together, but then we're multiplying those by the relative infestation threat to then get to our overall composite relative risk index. And we'll break down what feeds into each of these subcomponents in more detail going forward here. Here's a table from the paper which has way too much information to cover within a presentation, but just wanted to sort of illustrate another way of looking at this, all the components that feed into the model. In the first column here, we have those three different risk components, the impacts from infestation threat, or sorry, let me take that back. The very first component here is the likely, gives us an index of the likelihood of an infestation, that is the infestation threat. And numbers 2 and 3 here give us a sense of the impact that an infestation might lead to, what the value at risk is, and the additional spread threat looks like. Each of these is informed by at least a couple risk subcomponents, and those are detailed here, as well as the different data sources that we use to pull those all together. So overall, we use about a dozen different data sources, from USGS data sources to Army Corps of Engineers data sources, or NHD Plus data, and so on. So let's do a little bit of a deeper dive here into the infestation threat component. Again, this is the component that gives us a sense of what's the relative likelihood that a given huck would become infested. The way that we construct this reflects the idea that the probability of this kind of infestation is going to be a combination of the likelihood of an introduction and then the likelihood of an establishment after mussels are introduced. So if the habitat is completely unsuitable, then you can't have establishment and you would have no infestation threat there. So the first part here, the introduction threat, is given by a couple of different components. The first listed here is the distance-weighted sum of the water body access points to the nearest infestation sites. So this basically says within a given watershed, how many different boat ramps are there that give access to water bodies, and how far are those from existing infestation sites? A second component or subcomponent that feeds into the introduction threat is how much watercraft traffic do we see over land as identified by watercraft inspection and decontamination stations, or WID stations, and again, how far are those from existing infestations? So those two pieces come together to inform that introduction threat. And then the establishment threat is similar to what David had mentioned about using water quality to inform that. In our case, it's a bit simpler. We're using simply calcium and pH levels that are available across the watershed here to assess whether that habitat suitability in a given water body is low, medium, or high. And one other point to mention here as I present each of these components, you'll notice that there is a weight column here, and you can see just 0.5 and 0.5 here. As we were bringing these subcomponents together, we had to come up with some sort of way to weight them together in order to combine them to get to our overall component metric. These are subjective choices. Just as an example here, it's 0.5 and 0.5. But again, we might expect different managers or different decision makers to have different beliefs about what those weights should be. And our Shiny App tool allows the user to play with those weights to adjust them to what they think those appropriate weights are. For example, here on the relative importance of the introduction thread is given by boat ramps versus the introduction thread is given by overland watercraft traffic. So moving to the next component here, the value at risk. This essentially is a proxy for how damaging do we think an infestation would be if it was successful in a given watershed. To inform this, we bring together 4 different subcomponents. The first here being the water body beneficial use count. So this comes from EPA data that notes the different water beneficial uses that have been reported for different water bodies. And we add to that also hydropower facilities, which we think are likely important. Some of the other subcomponents here capture things like how much, what's the aerial extent of lakes and ponds, and what's the level of flow through rivers and streams, just to give a sense of the wetness of a given huck or watershed. How many water body access points are there, which gives us another sense of how interested people are in using the water bodies in that watershed. And also the watercraft traffic level, again, gives us another sense of how in demand access to those water bodies are. And again, we've got weights here in green, and for simplicity, they're all set equal, but of course, again, a user of the Shiny App tool can manipulate those and adjust those as they like. The final component of the spread threat, or sorry, the final component is spread threat, and it's comprised of 2 subcomponents. So you can imagine once a water body is infested, spread can happen either through natural spread. This is particularly important for, of course, rivers and streams. And so that natural spread potential is given by the downstream distance from the huck that we're looking at through the end of the Missouri River Basin at its confluence with the Mississippi River. So how much additional river and stream length extends from the huck itself, and that gives us a sense of the natural spread potential. And we also have, of course, the potential that humans could spread the mussels further, and we inform that through water body access points that exist within that huck. Okay, so at this point, I'm going to mention a couple of important details, but start to turn things over to Joe. So I will unshare my screen and let Joe start sharing. But as we do that, of course, as we bring these different metrics together, you probably noticed that these metrics aren't necessarily in the same units. Some are counts, some are have other units like kilometers squared or cubic feet per second. And so we've got to find a way to convert those into units that are more consistent with each other in order to combine them. So the way that we do that is we use we convert each of these variables into a percentile ranking. So for example, if there's a number of ramps within a given water body ramp or boat ramps within a given huck, how does that count of ramps compare to other hucks? What's its percentile ranking relative to those other hucks? And so by doing that, we convert what is a count measure, like the number of boat ramps, into an index that goes between 0 and 100%, or 0 and the 100th percentile. And that gives us a consistent set of units that hopefully leads to a more coherent way of combining these metrics as we look across all the different various heterogeneous metrics that we're bringing together. But that raises another interesting question, which is what relative comparison is most important? So you might imagine if you're a state manager, you might care about how any given watershed in your state ranks relative to other watersheds within your state. So we produce a version of the model that produces those percentile rankings based on just a within state comparison. But we also might be concerned about how these different HUCs rank relative to the Missouri River Basin as a whole, if you're concerned about this entire river basin and protecting it to the extent possible. And so we also have a relative ranking that ranks a given HUC relative to the entire Missouri River Basin. And Joe will give us a sense of how that matters for results coming up. So with that, let me turn it over to Joe.
Awesome. Thanks a lot, Mike. Can you hear me all right?
Loud and clear.
Nice. All right. Yeah, thanks, Mike. And yeah, thank you, Nicole and David, for your presentation earlier. So what I'm going to do is just follow up on what Mike's talked about and try to give you a sense of show you the results from this work. So to go off of what Mike was just talking about, the way we combine all this data is to use a relative ranking so that we can get an aggregate measure of the relative importance of each HUC, and the way that these weights can be adjusted in a way to reflect managers' priorities. So before diving into some of the results, just kind of to recall on the overall goal here is we're trying to see the relative threat from both quagga and zebra mussels, and to try to take into account as much data information as we can, and see all these categories of threats, which I'm going to go into in a sec, and to try to do so in a way that is relatively flexible, so that if a manager wants to prioritize one type of threat, so for example, maybe someone is more interested in spread threat, or just like the value at risk, like what Mike was talking about earlier, they could do so they could adjust that relative importance through our Shiny tool that we have. And so to give you a sense of what some of the data looks like, here is one of the subcomponents that we have. This is the boat ramp subcomponent. So this would be one of the most the least aggregate versions of the data. And so this is a map of the index that we produce for the boat ramp subcomponent, which would go into the value at risk index. And the green and yellow shaded watersheds, the HUC-10 watersheds, they reflect the raw data, which is the boat ramp locations. So these dots overlaid on the watershed maps represent boat ramp locations, and this gives you a sense of how we constructed these indexes. So we can see that there's a lot many boat ramps or water body access points across the Missouri River Basin, and watersheds that have, in this case, watersheds that have multiple boat ramps in them receive a higher weighting, and they get an index closer to 1. So naturally, this is generally how we construct these subcomponents, and then also to give you a sense of why we're combining so many of these components. One of the reasons is that we want to take into account as much information as we can, and this map of the in general, like the overall water suitability for the entire Missouri River Basin is, as Mike was talking about earlier, this gives us a sense of like how how suitable any watershed is to a new infestation based off of just calcium and pH. And the reason why we want to do this is that even if a watershed is very has a lot of high uses, so maybe it has a lot of local recreation potential that would be spoiled by a zebra infestation or a mussel infestation, we need to take into account that maybe the water is just not suitable for it, and so then a manager would not have to worry so much about protecting a certain watershed, or maybe they'd have to worry a little bit less about that. For our case in the Missouri River Basin, it's generally very suitable according to our metrics, and with the exception of a few watersheds in the most western parts of the Missouri River Basin, but these watersheds still come into play. So if that is to say that the ones that are shaded purple or green in this case receive a little bit less priority from our overall results. And this will actually end up becoming a relatively important factor. So this is our aggregated CRR composite risk measure that we have, and so this is kind of like the main result. And the idea here is to give a relative ranking of all of the watersheds from a threat of mussel infestation. And this map uses ranking for the entire basin. So it uses an MRB wide or entire basin wide ranking. This is in comparison to I'll flip back to it. This is in comparison to a statewide ranking. So Mike had mentioned earlier that we have two ways of comparing those two of indices. So in the cases of a MRB wide metric, we are interested in saying, okay, how important or how at risk is any given watershed relative to the entire MRB basin. But with this statewide ranking, we're saying what is the relative threat for watersheds only relative to the other watersheds in that state. And the reason that we want to do this is we want to be able to compare. We would like to be able to compare the differences between an MRB-wide focus and a statewide focus. And there are a few reasons for why we want to make this comparison between the MRB-wide and statewide results. One of them is that there are potential benefits for states pooling their efforts. So I'll go back to the MRB-wide results. So if for example a watershed in North Dakota gets infested, that becomes a problem for everyone in South Dakota, where those mussels can just spread and flow downstream. Additionally, if any state, so if I'm in Nebraska and maybe there's a new infestation near the border of Colorado, that then becomes an issue for the managers in Colorado, because now there's infestation that much closer to their state that could be transported via someone just moving their boat from one lake to the other across state boundaries. So that's why we want to be able to say, okay, can we compare what are the potential, or what are the differences between a whole basin-wide focus and a statewide focus, to see if there are maybe some potentials for cooperating cooperating on prevention efforts. Because mussels don't care about state lines, and yet boaters frequently travel between state lines. So this map is basically showing the difference between the MRB-wide results, so the whole basin-wide results, and the statewide results. So a green shaded huck in this case is indicating that the entire basin ranking is higher than the statewide ranking, and a purple shaded watershed is indicating that the statewide ranking is higher than the overall MRB wide ranking. Excuse me. And so some of the main takeaways from this, or some of like the main observations that we had from this from these initial results, were that, well, there's a lot of watersheds that are lit up green in Missouri, and that is basically reflecting that there are, from the whole basin wide perspective, these watersheds in Missouri, so like the easternmost part of the entire basin, they are more relatively important to the entire basin because that's kind of like the front line of infestation. So if you're interested in containing those infestations, it might be good to allocate more resources there to stop them from spreading further away. We also see in the eastern parts of Nebraska and the watersheds in Wyoming and Colorado, there's also some green watersheds there. That is reflecting the fact that those watersheds are rather upstream. So if those watersheds gets infected, we can expect infestations to flow downstream into Nebraska and parts of Kansas, which would further the spread. And then in North Dakota, we have an interesting case where North Dakota is very close to some infestation sites in our database, and we rank, or we weight one of the key parts of the model is saying, well, how close is a boat ramp to the nearest infestation site? Because that represents a potential node for a new infestation. And the North Dakota happens to have a lot of boat ramps relative to the entire Missouri River Basin, and it's relatively close to a couple of infestation sites. So relative to the entire basin, it tends to have a little bit more importance than it would otherwise if you were just a state manager looking at it. And then Montana is an interesting case where it actually gets a little bit less weight, because although it is almost the furthest upstream, it's relatively far away from infestation sites or known infestation sites. And so the relative risk compared to the entire basin is a little bit lower. Now, these results are the baseline results that we used with just equal weighting. So every subcomponent gets when we combine all the subcomponents, they all get weighted equally. But we want to be able to provide a more flexible means for combining the results, and we do that by offering this our Shiny tool, where managers and users can input their own weights to see how the results would change and interact with the data a little bit more. And so the idea there is, that someone could just, interact with the data. And if they, for example, don't really care about spread risk, they could just set that weight to 0, and the tool would automatically account for that. And then lastly, I'll just kind of quickly mention our ongoing work. We're looking at extending some of these results by doing some similar work for the Idaho and Washington areas, where we're using a little bit more granular data and looking at more different types of restrictions. So instead of just looking at a statewide focus, also considering what if you had watersheds that are disjointed. So in the example is like the Forest Service area that where your areas of interest are disjointed, but you still want to compare what like a statewide, an entire basin-wide approach versus those disjointed watersheds would look like. And using kind of the flexibility of these weights, we'd like to incorporate this part of the model into a network analysis that revolves around policy decisions like where to put a wood station. So with that, I think I'll wrap up and thank you for your time. Sorry for going a little bit over. Apologies on that. I hope we're not too short on time. And yeah, if happy to answer any questions.
Yeah, we have plenty of time for questions. Thank you, Joseph and Michael, for that wonderful presentation and really cool tool. So that concludes our presentations for today. We're going to move on to answering some questions. As a reminder, you can put your questions in the chat or in the Q&A. Sorry for that. It looks like Michael put the article from their work and the Art Shiny tool in the chat if you're interested in that. I'm going to check the Q&A. Looks like we do have one here. I'll share my thank you slide. Thank you, presenters. Your talks were wonderful. Looks like we have one question in the chat for Michael and Joseph. Is the R Shiny tool only applicable to the upper Missouri River Basin, or is it applicable to other areas in the West?
Yep.
Yeah. Right now, it's just for the Missouri River Basin. However, we're currently working on at least conducting similar analyses for the Idaho region and the state of Washington, which would include basically the entire Columbia River Basin. That is, we're not planning to release a public. I don't think we're planning to release a public tool for that just yet because it's work that we're doing with the with Washington Fish and Wildlife, like for them specifically. But that is something that is definitely possible in the future. But yeah, for now it's just the public tool is just for Missouri River Basin.
Very cool. Yeah, it seems like there might be few changes that need to be made to match the landscape in those areas, but yeah, hopefully available in the future. Cool. Thank you guys. While we wait for some other questions to roll in, I did have a question for David.
Yes.
I know, well, Michael and Joseph said they used pH and calcium to determine mussel suitability. Is that what you used? I think you mentioned that as well. Were those the two factors?
Yeah, I used, those were some of the factors.
Okay.
We used several other water quality parameters such as temperature, conductivity, but that doesn't necessarily mean Joe and Michael were off base because if I could only choose two environmental variables to use suitability for zebra mussels, I would pick pH and calcium. But yeah, we did incorporate several additional parameters as well.
Wonderful.
Yeah, I think this kind of highlights what David was saying about the value of having these water quality metrics available. For us, for the entire Missouri River Basin, we needed something that didn't require new data collection and was something based on existing measurements that were available to us. And so we kind of forced in that case to narrow down to the two. And then as you kind of narrow things down and or add in additional data collection to it, you can expand the set as David was looking at. But again, it just kind of underscores the helpfulness of having those water quality metrics available.
Wonderful. Yeah. Oh, looks like one just popped in here. One question from the Q&A, is there transferability of these tools to golden mussel out west? I know there's a lot of talk about golden mussel right now, so maybe anyone could speak to that.
I'm not super familiar with golden mussel, but as I talked about in the ending slide, there are many invasive species that are spread by the overland transportation or recreational boating. So the tools I was talking about, gravity model and graph theory, can be easily applied to those tools. Basically, the biggest change would be the environmental suitability, because obviously that would be specific for each species. And that is what would be changed out. But besides that, as long as the vector of overland transportation or recreational boats is the main vector of spread, then the tools I was using would still apply.
Yeah, and I think I would echo that. The whether or not the vector of spread is similar would be obviously key, as David just said, and then also if the style of impact is the same, or if there's something very different about the sort of damage that the species does, then we'd want to kind of adapt things to incorporate that. But I think the framework that David presented and the frameworks that Joe and I were looking at, these are adaptable to different species for sure.
Wonderful. All right, let me just check the chat and Q&A here. People are awfully quiet today. No.
But I think this was just another comment sort of on David's presentation against juxtaposed with ours. I think this is really nice. Like one of the questions we faced when we first were considering how to approach analysis for the Missouri River Basin was networks are obviously key here for both natural spread and especially human spread. And then there's also this massive abundance of data that you'd like to be able to incorporate. The tools that we have to analyze networks get a bit computationally intensive, and then so it's a bit hard to incorporate lots and lots of different data streams. So there's this sort of trade-off. Do you want like the sophistication of a network model to do network style analysis, and or do you want to try to do justice to all the different dimensions that you can inform with different variables? So where things are at with these kinds of approaches right now, there's sort of a trade-off between these two approaches. So I think this was sort of just serendipitously a nice example of those two different ways of approaching it. And going forward, we have some interest in trying to figure out how do you bridge that? How do you try to increase the data richness of the network methods? Or conversely, if you've got this data rich approach like the one that Joe and I presented, how do you bring network style analysis to that? How do you bridge these two different styles of approaching the risk characterization? So I think this was kind of a nice example of those two emerging prongs of the literature, and hopefully we're able to merge those in the future at some point.
Just to echo what Michael was saying, for my project, we had to make a simplifying assumption. You might have noticed I never talked about mussels spreading any way, but with the overland transportation of boats. Obviously, they can also spread naturally by river currents. But this was a master's project, and we just didn't have the time to try to incorporate that level of spread within the framework. So I really enjoyed seeing Michael's and Joe's presentation today on their methods that was able to incorporate that natural spread into their modeling of zebra mussel spread. But we're always trying to figure out better ways to improve our tools and incorporate these different elements that we weren't able to incorporate before.
Yeah, and I kind of go off of that and said, yeah, we also made we made simplifying assumptions even on the spread threat on that on that. For us it was more of a I'm not by training I'm not a hydrologist or an ecologist, so I know that from talking to folks that mussels can spread downstream, but how they do that and the rate at which that happens, and are there factors which could break that chain? Those are very complex problems, and for us we just said, okay, well, in order just to be able to provide some information, it's helpful just to say, well, is a watershed upstream from another or not, and say like, okay, that is some information better than nothing. But yeah, definitely there's so many ways to add complexity, but and realism to it, such as adding a little bit more, adding downstream spread mussel spread by just natural flow. Those types of models would be really interesting. But yeah, the difficulty definitely ramps up pretty fast.
All right, wonderful. I'm not seeing any more questions in the Q&A or the chat. Sam, you can let me know otherwise, but with that, I think we'll close our presentation, our webinar for today. I did just want to give a huge thank you and shout out to both our all of our presenters and everyone who attended today. As a reminder, the webinar is recorded and we will post it to the IMC's website. If you have any additional questions or follow-up comments, don't hesitate to reach out to us. And yeah, thank you again for attending, and we look forward to seeing you on the next one.
Thank you.
Thanks all.
Thanks a lot all.